Hello everyone, and welcome to your continuation of Chapter 4, where we'll be discussing Archaea and also some taxonomy. Whoop, went too far. So this is section 4.5, and we're going to make sure that you can list some differences between Archaea and bacteria. We know that Archaea, even though it was discovered close to when bacteria was on our planet, it is still more closely related to the domain Eukarya than it is to bacteria. One of the theories that are out there is endosymbiosis, and this is the thought that a bacteria cell was put and somehow got stuck into a Archaea cell. And so what happened is that kind of evolved into what we know as our eukaryotes today. Archaea and Eukarya tend to share some ribosomal RNA sequences that we don't find in bacteria. And instead, some of the ribosomal RNA in our mitochondria is similar to bacteria. So that's kind of where the thought of this endosymbiosis theory came from. And we also see that protein synthesis and ribosomal subunits, as far as their structuring, is similar to eukaryotes as well. Some differences from other cell types too is that Archaea have certain genetic sequences that are only found in their rRNA, and they have a unique method as far as their DNA compaction. And they have a unique set of membrane lipids and cell wall composition, which is what you see over here. And also they have pilin proteins. And a lot of microbiologists find these proteins more within bacteria. So maybe there's some relationship or overlap there. So here's the table that I told you guys to focus on as far as comparing and contrasting bacteria, Archaea, and Eukarya. We know that bacteria and Archaea, we classify those as prokaryotes, which we don't really use that term anymore. We just know it really means that they both do not have nuclei, and Eukarya are classified as eukaryotic. And our chromosomes are going to be similar between bacteria and Archaea here. So they are single and have a more circular nature to them. Also, like we said, they have no nucleus, so Archaea will also have its chromosomes within a nucleoid region, whereas in our Eukarya, we're going to have several, and it will be more linear. We've mentioned the ribosomes beforehand, but just as a reminder, Archaea and bacteria are both going to have the 70S units of our ribosomes, although we are seeing that the structure of it is similar to the 80S ribosome that Eukarya possess. As far as the flagella in bacteria, they are more hooked and ringed and have a hollow filament, whereas with Archaea, they're more solid fimbrial-like structures. And then in Eukarya, which we'll discuss more of in Chapter 5, they have a 9+2 microtubule arrangement. When we look at the ribosomal RNA sequences, we see that bacteria and Archaea and Eukarya all have unique sequences. And our bacteria cells are going to have just one sequence that is shared with Archaea. Archaea will have 3 sequences, but of course Archaea will share all of the same number of sequences. When we look at the protein synthesis, we see that bacteria shares no similarity to Eukarya, whereas Archaea does share similarities. And when we look at the peptidoglycan in a cell wall, only bacteria has this, which is why we can use gram staining for these cells. But if we try to stain Archaea or eukaryotic cells, then we will get nothing dyed. This is also why we can't do gram staining on our human cells. That was a part of your lab questions. And when we look at the cytoplasmic membrane, we see that we have fatty acids with ester linkages within bacterial cells. And in Archaea, we have long branched hydrocarbons with the ether linkages, and with our eukarya cells, we have the fatty acids with the ester linkages. And in bacteria, we don't have too many sterols within the membrane, with some exceptions, the exception being that mycoplasma that does not even have a cell wall. And within Archaea, we don't see that. And eukarya, we do have sterols in the within the plasma membrane. I don't know what happened to my slides here. Okay, so right now we think of Archaea as being the most primitive of all life forms, although there was some debate as to what came first, bacteria or Archaea. But we see that they originated about 4 billion years ago, and they tend to live in extreme environments that were found anciently, before we really had the planet Earth that we know today. So in areas where there are extreme heat, salt, acid, pH, pressure, or atmosphere. And we have classified them into 4 separate categories. So we're going to start first with our methane producers, which we're going to call methanogens, and they work to convert carbon dioxide and hydrogen into methane gas, which they thrive off of. And the common inhabitants of anaerobic swamp mud are areas that they would be found bottom sediments of lakes and oceans, and also the digestive systems of animals. So we even have some Archaea, some methanogens that can be found within our colon. We have also found some Archaea in the area of the vagina, and so we are slowly starting to discover more and more Archaea that can live within our bodies and also could cause disease. Gas will also be produced in the swamps and may become a source of fuel for these methanogens, and they may contribute to greenhouse gases, which keeps our Earth at the temperature that it is, and also global warming. The other category is going to be extreme halophils. So these Archaea are going to require a lot of salt in order to grow properly. If they don't have the proper salt environment, then they cannot thrive and reproduce. So they can multiply in about 36% salt that would destroy most cells. Sorry about that. I thought I turned this to silent. So we're going to find them in areas like inland seas and salt lakes, salt mines, and in salted fish. So this is a body of water here in which we can see the color changes, the red pigment, in order to synthesize ATP in the presence of light. And also in the bottom picture you could see some Archaea and the variety of shapes that we can find it in. Next we have the category of Psychrophiles, and these are going to grow at really low temperatures. So the picture to the right is some Archaea that we would actually find on different planets such as Saturn, Jupiter, and Mars. And then next we have our hypothermophiles. So this is the exact opposite. They are found to grow at very high temperatures. In fact, they flourish when they're in between temperatures of 80 degrees Celsius and 113 degrees Celsius. And we've found that in about 50 degrees Celsius, they stop having the ability to grow. And so we could find these Archaea in volcanic waters and soils and submarine vents as well. And usually we can see that salt and acid is tolerant as well as heat tolerant. Next we want to talk about identification of Archaea. A lot of the Archaea we cannot culture, and so we have been using our RNA sequencing in order to ID them. This has really advanced all of the understanding of Archaea and also the relationships between Archaea and other microbes, and has further our understanding of transcription, translation, and cellular evolution. So I want you to go ahead and do your concept check. How are archaeal cell walls different than bacterial cell walls? Define the terms methanogen, halophile, psychrophile, and hyperthermophile. And true and false, Archaea do not cause human disease. So for the next section, we are going to talk about some classification systems for bacteria and archaea. We'll talk about some of the Bergey's manuals that people have utilized, and also the 4 divisions of bacteria and archaea, and also define species in terms of bacteria. So our classification systems, we use them for both practical and academic purposes. We are going to be able to differentiate and identify unknown species, and also study the relationships and origins of microbes. So the characteristics that they used in order to come up with these manuals is that early bacteriologists were going to classify it based on shape and arrangement, their growth characteristics, and the habitat in which they live in. But now we have newer methods, newer technology, so we use a lot of biochemistry, genetics, and molecular traits. And we also are going to study the evolutionary relatedness that will most accurately determine them through comparison of nitrogen bases in RNA. So we've mentioned RNA quite a bit, so I just wanted to refresh your memory or let you know, in case you didn't really study cell biology prior to this class, that ribosomal RNA are those 2 subunits that come together. In our cells, they help with protein synthesis, and its genetic makeup in here, that RNA, is made up of nitrogen bases. So those bases, we have 4 of them, would be guanine, adenine, cytosine, and uracil. And so we can study those patterns and compare them to other microbes or other species. These are going to be pretty stable over long periods of time, and that's why we utilize them as kind of the gold standard of being able to classify these microbes. Any major differences that we see in the sequences is going to mean that we have distinct ancestry. And so you saw this chart before in chapter 1. This chart has been created based on evolutionary relatedness, and we link these 3 domains, bacteria, archaea, and eukarya, as coming from LUCA, one universal ancestor that they are all connected to. So we have only found cells or microbes as old as 4 billion years ago, and we are seeing that there is more of a relationship between eukarya and archaea. So here is Bergey's manual. This one was published based on our bacterial and archaea classification, and we used phenotypic traits, meaning we're using their shape, their culture behavior, culture thinking, how we create cultures, and also the biochemical reactions. We are still going to use this for our clinical microbiologists and researchers in order to quickly identify unknown bacteria. And then we have Bergey's Manual of Systematic Bacteriology. This is going to be more of a relatedness between bacteria and archaea, and is combining that phenotypic information with our RNA sequencing. And this is now available online. In case you're bored, you can always kind of venture on over there to look at some classification of bacteria and archaea. Next, we have Bergey's Manual of Determinative Bacteriology, and this is entirely based on phenotypic characteristics. They created categories of bacteria by the traits commonly assayed in the clinical realm, teaching, and also research labs. And we use this most for our microbiologists who are going to need to identify bacteria, but also want to know their evolutionary backgrounds. And this is also useful for students of medical microbiology. So our taxonomic scheme is going to use 4 major divisions of bacteria and archaea. We have our gurillicutes, which are going to be the gram-negative cell-walled and thin-skinned. And we have our firmicutes, which are going to be the gram-positive cell walls and thick and strong bacteria. And then we have our tenoricutes, which are going to lack a cell wall and be more soft, like our mycoplasmas. And then we have our mendocutes, which are going to be the archaea on their own. So we can also set up a diagnostic scheme and divide out our microbes based on whether they're gram-positive, gram-negative, or a type of bacteria without cell walls. And so we look at our subgroups of the cell shape, the arrangement, and then how they use oxygen. So a microbe that is aerobic means that it uses oxygen in metabolism. Anaerobic means that it does not use oxygen in its metabolism, and facultative means that it may or may not use the oxygen. So here are some medically important families and genera of bacteria. This is what you're going to use clinically. So as I mentioned in class, I will be creating a Quizlet stack of the ones I want you to memorize for your exam, and that'll be available under the Week 2, Chapter 4 tab on Blackboard. So the last thing we're going to talk about are bacterial species. And we know that the term species is the most descriptive type of taxonomy that we can have for a microbe. And so it is as a reminder, collection of bacterial cells that are going to share an overall pattern of similar traits. But we know that even though certain bacterial cells belong to the same species, that they are still going to have some different characteristics. And so we've created these subspecies, strains, or types in which will help us to differentiate and communicate that to our patients or to colleagues. And the other type is going to be a serotype, and this is based on how your body is going to respond to distinct surface molecules on the bacterial cell and the fashion in which your body uses its antibodies to respond to it. And so that takes care of the last part of Chapter 4. Please continue on to do the concept check number 6 and email me with any questions that you may have.